So this is our fourth part of the cell structure lecture. Here we want to talk about what we call the cytoskeleton of the cell. This is going to be the structural components of the cell that give it rigidity, support, enable movement. Think about all the features and functions of the skeletal system of the human body. Every cell has a comparable skeletal system. We just call it the cytoskeleton because it is the skeletal system of the cell. So it's microscopic. It's usually embedded within the cell membrane, the cell wall of plants and certain organisms that have cell walls. And it is composed of proteins, different protein structures. Now the cytoskeleton typically comes in three different shapes and forms. The filaments that make it up are the actin filaments, which are going to be the smallest, thinnest, and lightweight, or the lightest in weight and structure components of the cytoskeleton. These filaments, they're all protein filaments. They look like these big long strings of filaments of proteins strung together in a very simple basic protein form. So being small, they don't have a lot of strength to them, but what they do is they enable flexibility, kind of a cell contraction or cell movement with these actin filaments. Now in areas where the cells need a little bit more structural strength and integrity and durability, they will have things called microtubules. These are also made up of proteins, little amino acids. But the amino acids create a very thick tube-like structure. That's the middle structure here you're looking at on this particular slide, the microtubules. So that tube has more rigidity to it. It's the largest of the cytoskeleton structures. It's going to provide a lot more strength and durability to that aspect of the cell. A lot of times microtubules are used as little railroad tracks or trails or rails or whatever we want to call them that allow structures within the cell to actually move along them. So you think about a train. Train runs on that track. Cell structures like the lysosome have to move around, and the peroxisomes and the different structures that move, they move on these little tracks, these little, if we want to call them train tracks, within the cell. Those typically are the microtubules. So if you look at the bigger picture of the cell structure and the cytoskeleton there, you can see several green lines, green tubes running through it. Envision in your head those being little railroad tracks that different structures in the cell can move along. So it can go from one side of the cell to the other by sliding along these little microtubules. The third type of cytoskeleton structure are called intermediate filaments. Now the intermediate filament is also made up of proteins, so amino acids wrapped together to create a protein tube. And then there's multiple of these little protein tubes wrapped together to create the intermediate filament. So they're bigger than the actin, but smaller than the microtubules. So think about a cable. If you guys ever looked at cable or electrical wires, that's somewhat what the intermediate filaments resemble, is an electrical wire. More strength, more durability than actin, but less strength and less durability than the microtubules. So they're in between, which is why they're called intermediate filaments. A lot of times these will be woven into different parts of the cell membrane and used to help provide some structural stability for the actual cell membrane. So if the membrane's under pressure, gets tension and pressed upon, a lot of times intermediate filaments help with shock absorption and just that structural integrity. So again, you look at the bigger picture here, you can see a lot of those little kind of orangish tan intermediate filaments spreading through the cell membrane there, like a kind of a grid of filaments to help support it. All right, so when we look at the cytoskeleton, it provides shape, movement, structural support, integrity for the cell itself. So the cell and cell membrane, it has to have support, and that's what all these skeletal structures do for the cell. There are also structures called centrioles. Now this is your animal-specific structure, so make sure you guys remember that. This is found only in the animal cells. It's kind of a unique animal feature. Most everything else we talked about can be found in both plant and animal cells. But this one, this is ours. This is the animal feature that we do not see in plants. So this the centriole or centrioles are made up of microtubule triplets. So take a microtubule, group it together with another pair. So you have 3 total microtubules creating a triplet. And then get 9 of those triplets together. So a total of 27 microtubules, and you have 1 centriole. Now, during cell division, the process of cell division and mitosis, we will see 2 pairs of microtubules form. So they don't form until the cell says, hey, it's time to rip our DNA apart. We'll be talking about this later on as we get into the cell cycle unit. This structure will come back. It's an important structure. It acts as an anchor during the mitosis and meiosis process. If that anchor doesn't hold, if for some reason that microtubule broke, it didn't form correctly, it failed, then the genetic material may not separate evenly, and that can lead to some genetic disorders. So think about Down syndrome, or what we call trisomy 21. An individual inherited an extra chromosome. Sometimes the microtubules don't hold in place, and too much DNA goes to one side of the cell. It doesn't divide evenly, and it can lead to those problems. So we will also see the flagellum and another structure called centrioles on various cells. The flagellum is a tail-like structure, produces a whip-like action. It is made up of microtubules as well. But this time, the microtubules are in pairs, or we call them doublets. So 9 pairs of microtubules. They're arranged in a circle or a ring, and then there's a central pair that acts as like an axle. So think about a bicycle tire with spokes. The outer pairs act as the rim. The central pair acts as the hub or the axle, and then you have these spokes going into it, and it produces the flagellum structure. Wrap it with a plasma membrane, and now you have a long whip-like tail that has strength, support, and the ability to move. So in humans, the only cell that has flagella are sperm cells. We'll see flagella on all sorts of other cells out there, though. Bacteria have them, plant cells have them, fungi, lots of cells have flagella. But in the animal world, sperm are the only animal cells with flagella. Cilia are very similar to flagella, except they're short. They're short, little stubby, hair-like structures that produce a sweeping action. So they're found in the respiratory tract. They'll help sweep mucus and debris down the respiratory tract or try to get it out of the respiratory system. And we will also see them in the female reproductive system. They help propel the egg from where the egg is caught, a structure called the fimbriae, and they push and sweep that egg towards the uterus during the female reproductive cycle. So we'll see cilia and flagella come back when we talk about genetics, we talk about meiosis, we talk about reproduction. Those structures are important. If you're not producing flagella as a guy, you're not going to produce healthy, viable sperm. That's why we see all these abnormal sperm down there. 2 tails, 2 heads, goofy flagella. They don't swim correctly with a flagella tail that is not formed correctly. All right, so cells have lots and lots of different structures and features to them. Table 4.3 here is a great table for you guys to take a look at and try to use to help figure out what's found in each group of cells, the prokaryotics versus the animals versus the plant cells. So I would definitely encourage using this table as kind of a shortcut to help get that information. Table 4.4 it's talking about how cells connect to each other. So when we look at the connection of cells, those cells are various cells are going to be connected together, creating tissues. There's three general types of connections between cells. Tight junctions are junctions between cells that pull them together and create somewhat of a sealed connection between two cells. This produces somewhat of a barrier. It blocks substances from passing through them. So think about your digestive system. You don't want nutrients leaking out. So it creates a solid barrier with the tight junction there, trying to prevent substances from squeezing and leaking between the cells. The adhesion junction, also known as desmosomes, these junctions are going to provide a little bit of flexibility of the cell, a little bit of movement. They're often found in areas where there's mechanical stress associated with the cell, but they don't produce a solid barrier. So they produce kind of a porous barrier. A lot of times these cells, when they create tissue, the tissue acts as a filter. So it allows certain sized molecules or particles to filter or slip between the cells the key thing is based upon the size of the particle and the size of the space between the cells due to the adhesion junction. And then the third type of junction is known as a gap junction. This is also called communicating junctions. These are literally little tubes that will connect one cell to the next cell. It allows diffusion of molecules to occur from one cell to the next cell to the next, and allows a rapid movement of molecules and ions typically to just zip right through cell after cell after cell in a very fast, efficient process. As long as the molecules or ions fit, they can zip right through. So we'll see gap junctions in cells in the heart where conduction of ions has to occur in a fraction of a second in order for the heart to beat the way it does. We often see gap junctions associated with neuron cells to pass information from one cell to the next in a very rapid, quick fashion. And in the plant world, plants have a cell junction called plasmodesma or plasmodesmatas. And these are literally little tubes that connect one plant cell to the next plant cell. So they're comparable to the gap junctions of the animal cells. And it's the same concept. They allow movement of ions or molecules or substances from one cell to the next without having to filter and work their way through the cell wall. So when a plant pulls in water or minerals or ions from the soil, that can just zip right through to the next cell, to the next one, to the next one, and be conducted a lot quicker by going through these little plasmodesmas versus trying to diffuse through cell walls and cell membranes. So all of these different junctions connect the cells to each other. That allows them to form tissues. So cells connected to cells form tissues. Tissues connected to tissues form organs. Organs then form systems, and so on. So when we look at the overall picture of the cell, sometimes if a single structure within the cell doesn't work, that causes the entire cell not to function correctly. If that cell doesn't function correctly, the tissue may not work. If the tissue doesn't work, the organ may not work, and it builds all the way up to failure of the system and sometimes negative consequences to the individual. So again, keep in mind the example of Tay-Sachs. Talked a little bit about that. The lysosome isn't working, so that causes failure in the neural cells. That causes neural systems to break down and unfortunately results in death of the individual. We'll revisit Tay-Sachs with genetics, talk about how you get Tay-Sachs, what are the odds you might be a carrier for Tay-Sachs, and what possibly we can do to screen for Tay-Sachs, and maybe even down the road, can we correct Tay-Sachs and eliminate that genetic disorder from the population. So cells are the basic unit of life.